Hello everyone, it's Miss Montesano again. Today we're going to talk about Punnett squares, which is something that we went over last year. We went over these type of Punnett squares quite extensively, and you should have a lot of practice. The monohybrid, where you're just looking at one trait, and you're looking at the mom and the dad of that trait, and what are the likelihood of the offspring. Remember, the offspring are always found in here, having that trait. We are also going to dive into things like dihybrid crosses. Remember, that's the bigger one, a little bit harder for everybody to figure out. And that's when we're looking at the probability of the offspring having two. We're looking at two different traits and what are the probabilities of the offspring having those two traits or the various combinations that can happen. So again, that's the dihybrid. And we're also going to look at the laws of probability. And that would be your rule of multiplication and the rule of addition. Once we talk about these three things, they will kind of help you do the assignment that goes along with this, which is the sickle cell anemia activity. All right, so let's go ahead and dive into Punnett squares. Here we have a monohybrid, so this would be a big P and a little p and a big P and a big P. We'd call this a heterozygote cross. And the way this works is, here's dad, the sperm, you'd have the big P and the little p. The parents always go, this again should be review, always go on the same side of the Punnett square, right? And they're always on the outside. And here's mom, big P, little p. A lot of you guys will ask, does it matter who's on what side? Should the mom be on the top and the dad be on the bottom? From what I've seen, I'm sure there is a hard and fast rule, but for now, we're not really worried about that. It's just that if you look, all of moms are on this side of it and all of dads are on this side. The genes of the mom and the dad don't get to mix until you get into the little inside of the Punnett square. Okay, so let's take a look. In this case, the mom gives her big P, the dad gives his big P. The mom gives her big P and the dad's little p drops down. Here, the mom gives her little p and the dad gives his big P. And here, you have little p, because both the mom and the dad give little p's. All right. So here we have 3 purple flowers and 1 white flower, because here the purple is dominant over the white, so you have the homozygous recessive expressing the trait, okay? And here we go. What we could talk about this offspring having a phenotypic ratio of 3:1, 3 purple flowers to 1 white flower. Or we could say it has a genotypic ratio of a 1:2:1. 1 purple homozygote dominant, 2 heterozygote purples, and 1 homozygote recessive purple. So there's different ways that we can express what the offspring are. For your test, you need to know that. You need to know, okay, we could say it's got a phenotypic ratio of 3:1. We could say it's got a genotypic ratio of 1:2:1, okay? This is showing you it in a percentage, 25 to 50 to 25, or a 1:2:1. Phenotypic, we could say it's 75% purple to 25% white, or a 3:1 ratio, okay? So a test cross, we use this word, and we talked about this again last year, is when we breed a dominant phenotype, but we don't know the genotype, to a homozygous recessive. Now, that might already just make sense to some of you that have really enjoyed genetics and remember this from last year. Why would we do that? Well, by breeding the dominant phenotype, we don't know if that dominant phenotype is, in this case, let's just take the P as an example because we were with that allele. It could be a big P, or it could be the heterozygote, a big P, little p. So it could be either one of these, big P, little p, or big P, okay? But if we breed it with a homozygote recessive, that's what we would call a test cross, we can figure out if there is a recessive allele hiding underneath the dominant gene, okay? How do we know that? Well, that's where suddenly the mailman baby shows up, okay? So is it big P, or big P, little p? Well, basically what we find, I'm sorry, I just kind of skipped forward, with the test cross is that if it is the heterozygote, big P, little p, then you're going to get some recessive babies showing up. Actually, you have a 50% chance. So I could be considered a test cross with Joe because I have the dominant, let's say if we simplified eye color to, which this is not true, but brown eyes being completely dominant over blue eyes, and those were the only two colors in the world for eye color, which we all know that's not true. Those of you with green eyes are saying, hey, what about me? So if we said brown eyes were completely dominant over blue eyes, I would be considered a test cross because is there a gene for blue eyes hiding underneath? My husband is blue-eyed, so I would be test cross with him, and I have two girls with blue eyes, so it's obvious that there was, oh, we've got a cat showing up. It's obvious there was a blue-eyed gene hiding underneath, but this is what you would see. These two right here are showing you where you would have half a 50% chance of having the recessive genotype showing up when you did the test cross with a heterozygote. So a test cross just helps you see if you have got a homozygote dominant or a heterozygote, okay? All right, so a monohybrid cross, this is simple, easy stuff. It is the inheritance of a single trait, okay? Such as flower color, seed color, okay? All we're looking at with monohybrid crosses is one single trait. Generally, genetics aren't that simple. We want to know more. Dihybrid is where we have two characteristics, okay? This is showing you. We're looking at the seed color. Is it yellow or is it green? And is it wrinkled or is it smooth, okay? We call that dihybrid cross. So what are the chances of having a yellow seed that is wrinkled? What are the chances of having a yellow seed that is smooth? What are the chances of having a green seed that's wrinkled, okay? So it's the combination of those things, okay? The monohybrid, we only have one characteristic that is tracked. The dihybrid, we have two characteristics. And this is the one that gets trickier for you guys because generally I talk about it in general bio sophomore year, but not to the extent I would like to. So you have not had the practice, all right? This is showing you with the monohybrid. I like this picture because it shows you where these genes go up here and these genes go down here. The dihybrid's not that. It's a same principle going on to set this up where all of the females different allele combinations and meaning how they would be sorted in the egg are down here, and all of the males or all of one of the sexes is on top. The genes don't mix between the male and the female until you get down here, all right? And what I'm going to do, I'm going to go through a couple